From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on June 20th, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 46th episode of Matters Microbial. Those of you who watch and/or listen and spread the good microbial word to others, many, many thanks for this opportunity. Last week, I attended the American Society for Microbiology's annual conference in Atlanta, Georgia, and participated in 2 podcasts in front of a live audience. First, I was TWIM adjacent and co-hosted Arturo Casadevall with Michael Schmidt, talking about fungi and Arturo's new book. As you can see, having a live audience was different from being in my office in front of a camera. Then, I was lucky enough to record an episode of Matters Microbial with Michael Schmidt about the antimicrobial effects of metallic copper. It was recorded on my birthday, and some colleagues brought me a version of a birthday cake, hostess ding-dongs, with candles and sang to me, seen here. It was quite an experience, and I am both lucky and grateful. A wonderful experience was running into Dr. Rita Caldwell at the meeting, who was the subject of a Lux portrait created by my wife, mathematician Dr. Jennifer Quinn. So I was beyond delighted to take a photograph of Jenny, Rita, and Jenny's painting of Rita. Please see the show notes for some great information about Dr. Caldwell, including her recent book about her life. The topic of microbial evolution has long fascinated me, and I have been a long-term admirer of Dr. Richard Lenski, who has been a major figure in this field with his long-term evolutionary experiments with E. coli. Please see the show notes for some wonderful reading. Dr. Lenski is a fine scientist and wonderful human being. I have learned personally that evolution in the bacteria that I study takes place in the laboratory and has already revealed some interesting observations. Truly, the hand of Darwin is on all that lives. So it's a real pleasure introducing Dr. Nkrumah Grant, a new assistant professor of microbiology at the University of Illinois at Urbana-Champaign, to talk about his path in academia and his quite impressive work on microbial evolution. Welcome to the Quality Quorum, Nkrumah. It's wonderful to see you again.
Thank you, Mark, for having me on your show today. It's wonderful to see you. Before I stepped onto that, I was thinking about when we first met in 2015. I was a new graduate student then in Professor Richard Lenski's lab at Michigan State University, and we actually roomed together during this edamame workshop. So it's quite fascinating to see how far I've come from that time to now, right? Junior scientist, just starting to assistant professor. So thank you for having me.
Of course, it really is a small microbial world when you think about it.
Yeah, absolutely.
So I think that the first thing that I like to do in these podcasts is have our various guests talk a little bit about the path of how they got there. And I believe that you have quite a story to tell, if you wouldn't mind spending a few minutes.
Yeah, most definitely. I like to always kind of like premise these type of conversations in saying that, I'm very, very transparent, and I love to, share my story, the good, the bad, and the ugly. And I think it's important for audiences to know that, right? Just because, there's no straight path towards academia or success more generally. So actually how I became a microbiologist, I was long fascinated by microbes. I remember the first time that I looked into a microscope in 5th grade. The teacher had to literally take me away from the microscope to take me outside to my mother, who was waiting for me after school. It was about 15 minutes I spent after school, after class had ended, looking in through a microscope. So I've always liked microbes, the fact that there's something existing, that we can't see and doing things and moving around. It was just fascinating to me. So that was 5th grade. However, I think over the course of time, life has happened, and growing up in the inner city, there are some changes and things that had occurred. And one of those was me. I dropped out of high school. After dropping out of high school and serving time in juvenile detention for some while, I was granted the opportunity to go to an alternative education high school. And while there, I was reintroduced to microbiology in a way. So I guess kind of like how the story goes. So around that time, so 2006 is when I graduated from the alternative education high school. And a couple years before that, actually, there was something going on with like the stem cell research, right? So I was really, really interested in reading about stem cell research and understanding that we can really manipulate cells to, do things that we wanted to do or to take phenotypes and characters that we wanted them to have. So that was already in the back of my mind, although I was doing things quite different from being a scientist, right? Well, in going to this alternative education high school, my science teacher, Mr. Painter, he showed a show. There was a show. And in this show, the question was, what would life look like if we were to take some of the parameters here on Earth? Maybe, the wavelength of the sun, or, there's multiple moons. The atmospheric conditions are a little bit different. What would life look like under these conditions? And for me, that fascinated me. And I took my interests with stem cells and my knowledge of microbes living in, these strange places, which I had already known, and said, hey, well, life may not exist out there in the world as we know it here on Earth. And if it did, it would probably be microbes. And if not, we can actually perhaps one day engineer them so that they can live in those spaces. So that for me really kind of like, I guess, centered me on microbiology as being a field, more so astrobiology, actually.
Did you know that particular series you're talking about, Alien Planet, is one that I watched with my children?
So I did not know that, but that's the exact show. Yes, absolutely.
So that takes us to completing your high school equivalent. And then It was time for college at a place where I was offered a job, by the way.
Okay. So Grand Valley, I'm taking it there, Grand Valley State University. Yeah, so my college career started a little bit different than that, actually, too. So I actually started at a community college. Mm-hmm. So I had no idea of what I wanted to be, right? I had no thought whatsoever. So again, I graduated from alternative education high school. And in fact, I walked into college, by happenstance. I had to use the bathroom, really. I was waiting for someone, picking them up from class. I had to use the bathroom. They were taking a little bit too long. I went to the bathroom and walked around and said, oh, this is what college is like, it's like, it looks like high school, right? And I said, I can do this. That's really what it was. Mm-hmm. And I ended up on that Friday going in and taking kind of like admission tests and registering and so forth, and started the next semester after that. So bathroom breaks are important, I will say that, definitely. So I started there and I did some time at Delta Community College. I was also in Michigan as well. And after earning my associate's degree, I transferred to Grand Valley State University. Now, when I got to Grand Valley State University, and I will say I was just named Distinguished Alumni in Residence at Grand Valley, so I'll be honored there in November this year. It's quite a journey. I'm very excited about that. When I did get to Grand Valley State University, again, I had this astrobiology, idea of a field in the back of my mind. But when you tell people I want to be an astrobiologist, they look at you like the guy on, I don't know, that old show that was on the History Channel, right? So kind of like, he's crazy or something, right? It really wasn't much known about that. That said, I knew what it is that I wanted to do. I knew what interests me, and I took courses that allow me to, get the foundation knowledge that I needed to actually pursue that as a career field. So that was that. In fact, I also had dinner with Neil deGrasse Tyson while at Grand Valley State University too, because of that. So it ended up working out in my favor. So after graduating from Grand Valley State University, I then applied to a couple of graduate schools and ended up at Michigan State. Now, quite frankly, Michigan State was the last school that I had applied to. And it actually came by happenstance. I wanted to get away. I wanted to go, elsewhere. So I didn't want to stay in Michigan too long. But I emailed a few people who I was interested in working with at Michigan State University, Hema Ruggiera, Kas Kashefi, and Richard Lenski. And because it was down the street, an hour away from me, I just jumped in my car and I went. And this was before I even applied to university. And when I got to Michigan State University, I met with all these individuals. And Rich, by far, he was just, just an amazing, gentle, kind, smart, knowledgeable person. In addition to kind of telling me about the research work he was engaged in his lab, he took me to a couple of seminars, introduced me to several people on campus, let me know about resources that would be available to me on campus. And for me, I had already felt like I was a part of a family, and I would be a part of a family if I went to Michigan State University. So ultimately, that inspired me or encouraged me to apply to MSU, and that's where I ended up going for graduate school. After graduate school, I of course, I've done a couple of postdocs. I did a 2-year postdoctoral research fellowship at the University of Idaho, way across the United States. Worked on a plant-associated microbe there and did some experimental evolution with that microbe. I guess we'll talk about that a little bit later. This is just kind of like an overview. I did that for 2 years. Went on the job market for a little bit, got a couple of hits, and decided to just hold off a little bit. Went back to Michigan State University for an additional year where I worked with Vibrio cholera, which actually became what I founded my lab on. So I'm glad I took that second opportunity there. So that's where we are.
It's kind of interesting about the individual, seemingly random choices that put you just exactly where you need to be. And that's what struck me. Now, you should know, since we're swapping Rich Lenski stories, that I had been working in industry for many years and I wanted to get out of it and get back to academia. And I really wrote him a fanboy letter about a senior level postdoc. And he invited me out for an interview. He was so kind to me, treated me like I was a valued colleague. And he did not give me a job, but I felt great having gone through the process. Mm-hmm. That's the kind of person that Rich is. And he gets all embarrassed when I tell this story, but I don't care anymore. The important thing is that it was a wonderful experience for me, even though I didn't get the job. And how often can you say that? Mm-hmm. So I am not surprised that you received the proper level of mentoring, because that's critical. Just as you talked a moment before about how many people have an idea about the path to microbiology or to a STEM field in general. And the fact is there are many different paths as there are different people's experiences. And that's vital for everyone to think, because you don't hear that often. What you hear about are people that are almost pedigreed. Mm-hmm, mm-hmm. And that's great for horses and some dogs, mm-hmm. but it's not good for people. And I want everyone to understand, and I misuse this expression a lot, but I believe it. I have found microbiology to be not a big tent, but a big petri dish.
Absolutely.
And there's room for everyone there. And I continue to believe it, and I will believe it until the day I pass on.
Yep, absolutely. I think we share that philosophy.
Absolutely. And it's, there's that saying about having luck, and I'm sure you've heard it, and I have no idea, because old as I am, I wasn't there, that Pasteur said, chance favors the prepared mind. Mm-hmm. And I think all of us are given all kinds of opportunities and we don't often perceive them. What strikes me about you is that you grabbed opportunities with gusto. Mm-hmm. And it's really paid off for you.
Yeah, absolutely. That tends to be me. I like to take risks, I guess, chances in some way, but they're calculated most of the time. And even those that are not, I know that whatever the outcome is at the end, I'm willing to accept that. And I thank my mother for that. Yes.
That's wonderful to say. By the way, my parents had no idea about what the science business was, other than the people that were, say, on PBS in white coats and horn-rimmed glasses. Mm-hmm. And that's what they thought science was. They had no idea. And I was very fortunate, again, coming from where and when I did, to run into people who took the time to, how do I put this nicely, put up with my rough edges.
Mm-hmm. I guess.
And help me.
I would say, no, I'm sorry to interrupt you. I mean, that's extremely important, right? So we all come to our profession with our identities. And I feel like the time that you cast a shadow over your identity, you cast a shadow over your light as well too. So I try to really, really bring me to the table at all times, right? Because it allows me to think more freely, allows me to be more accepting of others as well too. So it's very, very important.
I also want to say before we talk, I do want to talk about the process of starting up a lab.
Okay.
Because, but I think before we get there, we should probably set the stage and talk a little bit about, because I love it, microbial evolution.
Right. That sounds good. So microbial evolution, right? So even before I started graduate school, I remember having an experiment, or we had to write a paper at Grand Valley State University in my senior thesis class, right, in the last year. And essentially, my group, we selected the topic of speciation, right? What defines a species, and how could we perform an experiment? What kind of experiments could we perform if we were to, study speciation if we wanted to? And I was quite excited because before I met Rich, before I knew of his work, I actually designed the LTEE, right, in my head. Well, it was there in 1988, right? So, but I actually, wrote up an experiment that was literally the LTEE. So for me, I knew I would work with bacteria, and I knew that bacteria grew fast. I knew that, I already knew the basics of kind of like genetics and biology and what evolution is, right? So I knew that changes to the genome that are selected over time, and then those strains or those species propagate over time, produce offspring that then pass those traits down to the next generation. So I already had a good mindset of that. So I designed the LTEE in this class, and I got knocked off one point, I remember, because like I said, okay, I would define a new species as being some like 3% difference. That was like some arbitrary number. I didn't know that part, right? So I remember getting 1 point down. So what is experimental evolution? It can be performed with, things that grow relatively fast, right? Or, and I say that relatively fast. Bacteria, it can be performed with things like Drosophila, yeast. It can be performed with, computer programs and these type of things. Any type of agent that can pass on information to the next, right? And that we can store in the interim. So in the case of, the long-term evolution experiment or some of the experiments that I've done, we work with bacteria. We serially passage bacteria in some media, some growth environment, right, that we are interested in. This could be in the presence of antibiotics. This could be under different temperature regimes, whatever selection pressure that you want to, so long as the bacteria can grow in it. They consume all of the nutrients. We then take those bacterial cells. We put them in more nutrients. They perform this process again, and we can do this iteratively over time. Now, these simple organisms are ideal for these type of experiments because we could take those populations, and not only can we collect DNA from them and look at the genetic changes that occur to these populations over time, but we can actually take them and put them in the freezer. Or in the case of a computer evolution, we could take that floppy drive and save the data over time. And then we can retroactively look at the things that have occurred in those populations, whereby in the genetics, whereby phenotypically or morphology, which I've done work in that space as well, looking at different changes in carbon utilization, output of toxins into the environment, whatever it is, right? And understand the dynamics, evolutionary dynamics, right? So that is microbial evolution. Now, while in Rich's lab at Michigan State University, I used this system actually to understand or to address some of the questions that I was interested in. My baby project, my favorite one, again, I'm interested in astrobiology, was looking at what happens to a facultative anaerobe or a bacterium that is capable of using different modes of growth, metabolism to grow, right? So in the case of E. coli, it's a facultative anaerobe. It can grow aerobically in the presence of oxygen or in the absence of oxygen, right? And what I was interested in saying, in the LTEE, these populations have been evolving in the absence of oxygen for 35 years at the time. What has happened to their capacity to grow in the absence of it, right? That was kind of like my thoughts, right? And I already had a hypothesis. I thought that they would maintain this metabolism. Why? Because anaerobic metabolism had evolved 2.5 billion years before aerobic metabolism, just a simple hypothesis. So I felt like this would serve as a scaffold, right, for every other thing that grew on top, right? All these other reactions that emerged on top, right? I tested this hypothesis, doing a bunch of, genetic sequencing, actually competing ancestor and evolved strains in aerobic and anaerobic environments, right, where they're competing for these nutrients. And I actually found that not only did these populations maintain the capacity to grow in an environment they hadn't seen in 35 years, right, but that they also improved in that environment, right, suggesting that, yes, there was some kind of buttressing or some maintenance that was going on to maintain this metabolism.
It's interesting, if you don't mind my saying so, when you say 35 years, those are human years.
Correct.
And when you think about the number of generations that these bacteria had been under that particular regimen, it comes out to the equivalent of tens of thousands or hundreds of thousands of years.
Actually, billions, right? So if you think about the average, generation time, about 21 years or so for humans, right? So where bacteria, they're going through 6.6 generations per day. So in that study, for example, I looked at 50,000 generations of, bacteria, right? Which again is about the billion years of human evolution, right? So it's crazy, right? It's crazy.
You have, I believe you've met Bill Metcalf.
Yes, I have.
Right. And Bill Metcalf is full of pithy sayings, as you know, if you've met him.
Yes.
And I was doing a phenotypic assay. I was looking for things that turned McConkey maltose media red. And I was trying to complement a mutation, and then I knew that it had complemented it because the colony would be red on that medium. Mm-hmm. And then Bill was great because he said to me, bacteria are not in the business of following your phenotypic assays. They have their own agenda, and it's not yours. Mm-hmm. And I'm not trying to be vitalist with you, but it is funny how easily we get fooled when we think about that. We talk about the way that we grow microbes in shake flasks or in tubes or on dishes, and absolutely there are changes that take place with, and I use this term advisedly, domestication into laboratory conditions. But you have to start somewhere, is the point.
Absolutely.
And what I like is you can phrase a hypothesis and you can test it. It's very hard to do that with mice.
Mm-hmm, mm-hmm. Definitely. And a little bit more messy to do it with mice as well, too. I could take my microbe, I can, disinfect or sterilize and then toss it down the drain if I needed to, right? Where mice, I don't know the procedures there. The IRB approvals enough scare me, I guess. It's a little messy there. Yeah. Mm-hmm.
So this has been a wonderful project to find out that they maintained this particular, I'm going to call it kind of a subroutine. I don't mean to be computer sciencey with you, but this subroutine to survive well under anaerobic conditions, even though they hadn't seen it in the equivalent of many, 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 many generations. Mm-hmm. And this works counter to what most people think of with evolution, where if you don't use it, you lose it.
Absolutely.
And I believe that this buttresses something that I just mentioned, that there are often other things that those pathways are doing besides the business of surviving well in the absence of oxygen.
Absolutely.
And I tend to be hard on myself saying I'm not as smart as a microbe. And I think that that's part of this.
Mm-hmm. Definitely. There's definitely some. So if I looked at those populations, this is going to actually give perfect segue into the work that I'm doing now. If we look at those populations that, maintain the capacity to grow anaerobically, right, they all had a mutation in a two-component regulatory system. And these are systems that actually serves as switches. In this system, this is the ARC-A and ARC-B system, which responds to, the anoxia in the cell, right? So it can respond to that. And when conditions are that or oxidizing conditions, they perform then the anaerobic metabolism, and under reductive conditions, the opposite, right? So they all have a mutation in that system. And because of this, so this serves as the switch, they all have this mutation. What we believe, or what I believe is going on, I hypothesize, is that we see constitutive expression of genes that should only be expressed in the absence of oxygen, not being expressed in both environments, allowing selection to then act on those things simultaneously. And there's also some other like growth dynamics that are going on in those populations as well too. So by the fact that they're growing extremely fast, so microbes, they do something called oxidative. Well, essentially, let's reverse that. So microbes, they do something called overflow metabolism, right? And overflow metabolism is when instead of performing oxidative, respiration, they actually perform fermentation, right? Mm-hmm. And they do so because they are growing extremely fast and they're not producing enough of the cofactors, right? And the redox, right? So they have to just do that to actually dump off electrons. So that could also be a way that they're in the oxygenic environment actually maintaining some of the anaerobic machinery in the absence of growth for it. And we do know this because these populations, at least in Rich's system, they produce things like acetate, fumarate, succinate. These things are in the media that we can actually measure. So we know that they're doing the fermentation.
I hate to tell you, but this is so relevant to some things that I'm messing with in the lab myself that I need to talk to you about it another time. But thank you in advance.
Yeah,
you just gave me some great ideas.
Great, absolutely. I love that you have to tell me that. So what does this mean, right? So that was E. coli, and there's interactions that are occurring. I think that's the way to sum it up. There are genetic interactions, or, I like to think of a cell as an ecosystem, right? So I think we can apply ecological principles to an individual cells. You have proteins, you have flux, you have things going in, you have things going out, you have proteins interacting in time and in space, at coordinated points, right? And I think so, genetic interactions is really what interests me. So if we can go way back in time, right? We know that bacteria now, they can have hundreds to thousands of genes, right? But way back in time, there was some minimal set of reactions that was occurring, right? And over time, you get the addition of 1 or 2 or 3. Maybe you get a copy of a gene and then now diversifies and allows one to do one thing while the other one is doing what it was doing when it first arose, right? You have all of these things occurring at one time, but they don't exist individually. You make a mutation in one or you remove one, you change flux and you change the architecture of the genome. And that is really, really what interests me. And that's what my lab is working on with Vibrio cholera here at UIUC. And Vibrio cholera is a good model organism for several reasons to do that. So first and foremost, just a little bit about Vibrio cholera. It's a public health threat. I call it a disease of inequity. It infects about 1.3 to 4 million people annually, and about 21,000 to 150,000 individuals die from the disease cholera that it causes each year. Cholera is a extreme diarrhea, right? It's caused actually by a bacterial phage that is inside of Vibrio cholera itself. Causes profuse diarrhea. Up to 3 liters can be lost, right, in a matter of just an hour. There are therapies or intervention strategies that can, it is treatable. So typically patients who are sick with cholera, they receive rehydration therapy, which is essentially just sugar water and salts, right? So a lot of potassium, sodium chloride, and glucose of that nature. Cholera is why we can use it to study genetic interactions, because it readily picks up DNA from environment all of the time. It's what we call a naturally competent organism. It's always when in the presence of chitin or when in association with shellfish, right? These type of things, it actually takes up DNA where it can use it, as food, to some extent, and also can use it to acquire new traits from the environment, to shuffle its genome, right? And because of this, then we can actually begin to shuffle the genomes ourselves in ways that we are interested in, interested in and look at these genetic interactions. And I've developed ways to do that in high throughput here in the lab.
You know, it's a fascinating, okay, when I say a fascinating organism, I'm talking about a disease, and that is not fascinating, that's sad. But first off, just for listeners and viewers, I mean, you essentially die of dehydration, as you mentioned. You will lose, I want to say 20 to 40 liters per day of liquid.
Mm-hmm.
It's horrendous. The CTX gene, which is, as you say, carried by the bacteriophage, fascinating in that. I love the idea of mobile DNA elements that carry information involving the ability of an organism to interact. And what it does specifically, as you know, is it tickles kind of the system that balances salt concentration in our gut. So it fools the body into dumping liquid into the lumen of the gut, which is the source of the diarrhea. This presumably is creating this fantastic growth medium and ability to disperse itself.
Mm-hmm.
The chitin issue is especially interesting to me, but before that, one thing that I teach, and again, I'm sure you know this, is the business of when people are drinking contaminated water. If they are malnourished, it is much easier for them to get the disease. And that's because you have a heightened pH instead of a lower pH in your stomach and upper gut. So this takes it very much to the inequity issue that you talked about there. So food insecurity can make this much worse. Finally, the chitin is fascinating because Rita Colwell, who I mentioned before, was one of the people that showed that Vibrio cholera colonizes the surface of small crustaceans.
Mm-hmm.
And that's how you're able to get a high dose of the cholera organisms. So she had a wonderful finding that you could filter the water, excluding these small crustaceans, these amphipods, and therefore reduce the incidence of cholera. So there's a wonderful, as you say, a wonderful system. There's strong molecular genetics that can be studied. There's issues of social equity that can be studied. There's transfer to other types of cultures about... Whenever I use the word culture in microbiology, I laugh at myself. But
Yeah,
you know what
absolutely.
I mean, different
Yeah,
sociological ideas.
Mm-hmm.
And all of this works together. The fact that they're naturally competent to take up DNA is just icing on the microbial cake.
Mm-hmm.
So this is a perfect system. I can't wait to hear more.
Yes, absolutely. Good. So, yeah, so actually I could kind of talk about what I did in my postdoc leading up to my lab, because I think this introduces the importance of culture, right? Actual culture, not culture microbes. It's the same thing. So I won't laugh at myself. We've already done that. I think it actually emphasizes the importance of culture as well, and what, having diverse trainees actually brings to the table. Great. So I'm interested in genetic interactions. Vibrio is naturally competent, so I knew I wanted to work with them. So when I was in finishing up my postdoc at the University of Idaho, I got in contact with Chris Waters at Michigan State. I'm like, hey, I want to apply for, a couple of grants, but I need a mentor, right, to serve as my point, right, point trainer, I guess we can call it there. Would you be willing to serve that? I want to work with Vibrio, and here are some of the things that I want to do, right? So he said fine. So he was on my graduate committee while I was at Michigan State. So we had already had a good relationship, and that was good. Long story short, I did get some NIH funding, and I went back to Michigan State for a year. Now, my project, initially, I wanted to look at kind of like mobile genetic elements, phage actually transfer, and, taking it with the perspective that when we think about our issues with antibiotic resistance, now we're using phage as an alternative to that. Some of the same dynamics that we see emerge with the overuse of antibiotics. What is the potential for this to occur with phage? That was kind of like, what I wrote the grant actually for to do. However, in kind of like thinking about, some projects, a graduate student in Chris Waters' lab, Alex, he had been doing some experiments where he was creating a transposon mutant library, and he killed his library. And then he realized that, if I take Vibrio cholera and I induce natural competence, I could rescue this library, right? And that to me was just very, very fascinating, right? Because then I said, okay, the pool of mutants that you're getting to interact is going to change probably in that rescue, right? And if it changes, how much so? So, given that said, I said, all right, we knew that there was a library that was constructed, a transposon mutant library that was constructed by John Michelano's group, maybe like in 2008. And this is a non-redundant transposon library. And what do I mean by that? So transposons, for the audience, are just genes that jump around the genome and they disrupt the function because there's a big piece of, a sequence that just disrupts the gene. John Michelano's group, they had created this non-redundant library, which just means that every single gene has been systematically knocked out with a transposon, and then these mutants are then arrayed across the wells of 96 well plates, right? So about 3,100 different Vibrio cholera mutants, each with a unique gene disrupted in the genome. Now, unbeknownst to the Michelano's group was that in this library, when they went back and did the sequencing, they found that there was an off-target mutation. They made there was their parent strain in which they were putting these transposon in had a mutation in a gene called LUXO. And why is LUXO important for this story? So Vibrio cholera exists as, in a planktonic state and also in biofilms, right? High cell density and low cell density state. And these differences in transcriptional states actually dictate their ability to cause disease, their ability to move around, their ability to colonize surfaces, and all of these, and even their ability to withstand phage, right? Infection by phage. The mutation in this LUXO gene locked these populations in a low cell density state, which means that while this was a rich resource for discovery, for vaccine development, and for just understanding Vibrio cholera dynamics, including their mortality, it was kind of misrepresentative of actually how Vibrio cholera lives in the wild. Also in this story, Ankur Dalia at Indiana University, we know that, and actually like Melanie Blockish as well, has done some work in this system as well too, we know that Vibrio cholera is naturally competent in the presence of chitin, but we can bypass that actually by expressing a regulator of a couple regulators and some small RNAs, we can say, of the machinery that dictates its ability to become naturally competent, right? So the thought was that since Alex saw that we could rescue these libraries, we have a system to induce natural competence. We have this library of mutants that are now, that are in this low cell density locked background. If we take a wild type cholera, we add a plasmid that we can induce natural competence, we can get gDNA from these libraries and then co-incubate it with this parent wild type strain, this new wild type strain. We can then have a clean genetic background in which cholera now can switch between the low cell density and high cell density states. And we can then look at the effect of that single gene in a background that is representative of cholera, right? So that was kind of like the goal of my project. Now, again, there are 3,100 mutants, right? Now, we know that we can extract gDNA using kits, and that works, right? It worked perfectly. But I don't know how long it takes you or your students to do gDNA extractions, but I know for me, 50 extractions is going to take me an entire day. And it's timely. You have all of these spins, you have all of the decanting and, looking at the tube to make sure that your pellet is there. So much waiting, right? And it's expensive, right? So I wanted to develop a way in which I can actually extract genomic DNA in high throughput. And this is where the culture comes in. Right. So I did a couple of different things, right? So I said, all right, I just need to lyse cells, right, and get DNA. I can get DNA from bananas and strawberries. I've been doing that forever, right? Once it's there, I can do alcohol precipitation, phenol extraction, or something of that nature, right? I know that I can do that, but I need to be able to lyse these cells. I tried to freeze thaw Vibrio cholera cells so that they can lyse. That wasn't sufficient. I microwave cells so that they would lyse. That also didn't work too well. And I did a bunch of other things that didn't work out. And I was very, very frustrated. I think about 2 or 3 weeks into, my postdoc, like this should be very, very easy. And I didn't want to go the long route. But I have this very, very, very strange obsession, right? Every night before I go to bed, I read something random on Wikipedia or something online, just something random, completely outside of science. And I also love to cook. Now, I'm a first generation citizen, American citizen. My family immigrated from Guyana, South America in the 70s, and I was the firstborn here. So I have a lot of Caribbean culture, and I love to cook Caribbean food. Now, how does these stories mesh? Now, there was one time I was reading my random Wikipedia story, and I was thinking about a pot of food that we call pepper pot. And pepper pot is actually made from cassava that has been, boiled and stewed down. And we have a lot of poisonous with cyanide. Cassava actually has a lot of cyanide, right? So the Amerindians, or the native people of Guyana, they developed a way to actually cook the cassava, where they're actually sieving off the cyanide, and then they get this nice thick paste that you can actually make stews in. And what people would do with this syrup, it kind of looked like Korean barbecue, I guess, but it has a very, very unique flavor. They would take meats that they would go and hunt, from, the bushes in the Amazon in the area, and they would just add it to the pot. And my Wikipedia search was wondering, why doesn't this food spoil? They never wash their pot. They just add more of the casrip, is called, and more of the spices. And I found that there were a lot of things that were antimicrobial and antifungal in this dish, right? So in my frustration, I knew I needed to kill Vibrio cholera cells, and I knew I liked I knew that I liked to cook. I said, all right, let me check casrip Vibrio cholera, pepper Vibrio cholera, all these different spices, and I got a hit, right? Thyme and Vibrio cholera. There was a paper published in 1946, and the paper is a paragraph long, literally a paragraph long. And within that paper, the author said, he was from India, that if you take thyme, right, or thymol, the actual extract of thyme, and you add it to Vibrio cholera, within an hour, you sterilize the cultures, right? And I was like, you sterilize the cultures. I went to my local grocery store. I bought thyme. I bought mineral oil. I did a simple extraction on my stove, took that to the lab the next day, added it to a culture. And when I tell you within an hour, everything crashed out of that culture, it was beautiful. It was absolutely beautiful. So then, long story short, I then ordered the extract. It's called thymol from, I don't know, one of these companies, Eppendorf or Fisher or one of these companies. I ordered it from them and then used the raw material, had a salt, alcohol and salt precipitation technique to extract DNA. And I was able then now to develop a gDNA extraction kit that is about 64 cents per reaction, where the alternative or comparable kits are about $3.90 per reaction. Mm-hmm. And I can do as many as 500, 600 gDNA extractions in a single workday. And remember, I told you it would take me 50. So it was highly economical. Very, very efficient. And I was able to transfer this library into this new genomic background, right, in a matter of 2.5 weeks. So I think without the culture, it wouldn't have happened that way. I wouldn't have had that mindset to actually go about doing it.
Two different definitions of culture here, aren't there?
Absolutely.
And this, have you published on this?
Yeah, so actually, I have a paper on that in review currently. And my students, my 2 graduate students here at the university, they're actually working on a Jove article actually for high throughput gDNA extraction.
Wonderful.
So they're working on it now. It's actually due today. So, yeah.
And it's funny, there's a lot of information about the antimicrobial aspects of spices and foods, and it's something I used to teach in lab. There's even a paper about what's antimicrobial in salsa, which makes me laugh so much. I mean, it's like you're sitting around eating with chips. I wonder if this is antimicrobial. But you see, it's worth checking. And this is a great example of how everything came together. What a great project.
Yeah, absolutely. Yeah, it's very, very exciting. Because
So,
now I can take these different mutants, for example, these 3,100 mutants. I could take mutant 1 and mutant 2. I can extract DNA and actually combine those very, very quickly. So we're doing a lot of experiments like that now where we're actually making these multigenic mutants, we'll just call them, looking at these genetic actions.
You wrote a wonderful, or you were a co-author on a paper talking about different systems to study things. And many times on this program, I've talked about the dangers of eukaryocentrism. I've talked about the dangers of oxycentrism. But I would also say, and this, what you've just discussed for the last few minutes is very relevant, the dangers of colicentrism. You really have to quit assuming that all things are like E. coli or that all things are like what it is you've studied. You have to have an open mind, and that's true absolutely in science. And this is a wonderful example that I will be telling people about.
Yeah, absolutely. And I think it's kind of interesting because well, I just mentioned this gDNA extraction technique, right? So Vibrio cholera is a gram-negative organism, just as E. coli, right? But the efficacy of these compounds is actually different on both of them, right? So, we think this compound actually impacts the cell membrane. That's one thing that we like to say. That's what the consensus the field has come to. But if that's the case, then you expect for E. coli and Vibrio to behave, the same. But this is a good example, too, that what is true for E. coli is not true for, another species.
We had a fun conversation with Esther Angert on this program a couple of weeks ago. And she used this expression. I made it into a meme and I'll have to send it to you. And you know the famous Monod quote that what's true for elephants is true for E. coli. And what she used to say is what's true for E. coli is true for E. coli. And it's just kind of a joke because we get this into our head that that's the way all organisms would work. And I would argue that not all E. coli are the same either.
Yes. Yep. I would argue the same.
And we have this reductionist attitude that we want to put everything into I was at a research conference one year, and someone was talking about the temperature optima for luciferase from a deep ocean organism. And it worked, of course, better at colder temperatures. And a very famous person I will not name asked them, where is this organism from? And the person said, well, it's not from Sigma. And it turned into like a kind of an unpleasant difference of opinion. But the fact of the matter is that these are part of the natural world and we take it out and we cultivate it and things change. Now, what's good is your approach is you can actually take these individual hits in the individual genes that we know of in the reference genome, and then you can put them into whatever background you want. Because most people are unaware that when you do many forms of mutagenesis, you get second and third hits as well. You have to clean up that background, and that's laborious.
Correct.
And you just found a great way to do it.
Absolutely. A great, efficient, and economical way to do it.
And it's really good not just for the study of what genes are involved in issues involving the ability to cause disease. See, Arturo Casadevall is teaching me bit by bit not to use the word pathogen. So I've been careful. It's all contextual, and I think that that's true. But it's hard if there's a word you're used to give up.
Right, absolutely.
But this is like a really good system for incoming students. So what I'd like to do now, if you don't mind, is transition a little bit over to what it's like to set up a new lab.
Yeah, great. No problem. Setting up a lab has been, extremely rewarding and exciting, right? So again, like I said, you met me, we met each other in 2015, right? I was very, very young. And while I did have a good idea of what I wanted to do and where I would be, you don't know whether or not you're actually going to realize that, right? So I will say that it is such an honor to be here at the University of Illinois Urbana-Champaign, who also told me no when I was applying for graduate school. I will say it was the first denial that I got actually. So it's kind of interesting and fascinating that I'm here now. And I will say too that I think that a lot of the culture, human culture, we tend to complain a lot, right? So, and while there are some things I guess that are could be changed in academia, right? There's no job that I think that is as rewarding to be able to have ideas and follow through with them, right? And to train the next generation of students, right? To, inspire them in such a way that your mentors or my mentors, for example, have inspired me. So I absolutely love, being in the position that I'm in. People looking at me for answers, and if I don't have them, having ways to find them. And it has just been exciting. I guess dealing sometimes with the sales rep has not been too exciting, but I've learned to close the door and kind of hide out. Oh, did I say that out loud? I think so. Yeah, but oh, shoot. But yeah, so it has been fascinating.
Mm-hmm. So you don't have a lab website up yet, because I was looking.
Not yet. So actually, it's being made currently. It is someplace. I won't tell you where, but it is published in a way, but it's not complete yet. Maybe next week.
So what I like, and I could probably add that if you get done in the next week or so. I'd be happy to add that to the show notes, because
Absolutely.
what's interesting about a laboratory website, especially for folks at R01s, is there's usually a statement about what the philosophy of the lab is. There are pictures of the members. There's a description of the research projects you have available. There's so much that gives you a flavor of what it's like to work in a laboratory with a particular PI.
Mm-hmm.
I grew up at a time that there was a television show called Magnum PI.
Oh, I know that show.
Private Investigators. So when you're in science and you hear about PIs, it just makes me laugh.
Uh-huh.
But I'm really curious, what kind of students are you looking for in your laboratory?
Absolutely. So I really, really take mentorship seriously, right? So I think approach mentorship more so kind of like, in a lab, like it's a family, right? You will never disconnect from your graduate trainers. I mean, your graduate mentors, for example. I stay in contact with mentors from community college, some 18 years ago now, right? Just a very, very long time. So I take that very seriously. So what I'm looking for is people who integrate in a lab, right? And integrate in a family. I'm also looking for people who aren't afraid to fail, or let me flip that, actually try new things, right? So I think a lot of the things that I have done in the lab, I'll say, there was some serendipity, but there was also me just saying, okay, here's a new technique. I am going to, go and try it out. I'm going to see how I can use this potentially in my work. I'm also looking for people who are willing to, bring themselves to the table, who aren't afraid to do that. I think, you mentioned this earlier, a lot of times, there's a cookie cutter, right? Science, I think sometimes, or academia can make cookie cutter type individuals, and in doing so, you lose flavor, right? I definitely believe it. So I've given plenty of talks to high school students, to, elementary students as well. And I'm asking them like, hey, like, what is your impression of what a scientist mm-hmm. looks like? What do you think that we do, right? And I'm the last person they think of as a scientist, right? Until they see my work, right? And they realize mm-hmm. that is what it is. They think I'm a bodybuilder or some kind of coach somewhere or whatever it is. Just changing the dynamic requires that people who are bold, who are bright, who have their ideas, and who are willing to, go to lengths, to answer their questions. Those are the people that I am looking for to join my lab.
There was an expression here at my institution. We had a meeting about diversifying our faculty, and we had a consultant come and talk to us. And there's an expression that everyone's heard of by now in academia, which is safe space, mm-hmm. right? Mm-hmm. But this consultant said something that really galvanized me. She said that her goal was to create a brave space.
Mm-hmm.
And it sounds like that's what you'd like to have in your laboratory, is a brave space.
Absolutely.
A place where people show mutual respect, they celebrate their differences, and they listen to each other, respect one another.
Absolutely.
And I don't want to give the impression that that doesn't exist. It's often not codified. And again, this takes us back to the kinds of things that appear when people write about their laboratories. And this is a really good example of it. You have to have that brave space to actually work together. Some folks don't like the term that you have a family based on your lab. They go odd places with that. But my response is there's all kinds of families.
Absolutely.
Sometimes they're better than others, right? There's a famous Robert Frost quote. I don't know if you've heard it from one of his poems, that home is the place when you have to go there, they have to take you in. And when I was little, I read that. I didn't see kind of the hey, you have to take them in. But it's true. You owe them. I owe my students. And frankly, it doesn't matter what they think of me. It's my privilege and my responsibility to do my best for them. And I try my best at that. And I know you do too.
Absolutely. So I have a disclaimer. So when I do and speak with students, I tell them a couple of things. I say, I am friendly, but I'm not your friend. Right? I come out and I say that I'm friendly, but I'm not your friend. My objective, my responsibility is to make sure that I provide you with the training that you need in order to occupy any space that it is that you want to occupy when you leave my lab, whether that be in industry, whether that be in academia, whether that be, at a startup company, right? You are a reflection of yourself, and you're also a reflection of the training that you got here, right? So it is my responsibility to make sure, right, that you feel prepared when you leave my group, right, to enter those spaces. So I let them know. And each time I meet with them, I ask them questions like, okay, how do you feel? That gives them authority and ownership. How do you feel how do you feel the progress that you are making, right? How do you feel about the progress you are making, right? Is there anything that I can help you to meet your goals, right? What are your goals? I ask these kind of questions to make sure that they know that they're on task. It's their responsibility ultimately. So that's how I approach it.
I think this sounds wonderful. Nkrumah, it's been a wonderful time chatting with you. And I want to say, yes, we were roommates at Amami. And yes, you brought me a book from Rich Lenski and all the rest. I have been so delighted at the path that you've taken. I've been so happy to see you develop into the mentor and scientist that you are. But I had no doubt you would do those things, even when I met you all those years ago, almost 10.
Yeah.
The fact of the matter is that you really do walk the walk and talk the talk, and you are an exemplar of what I would see for the future of microbiology.
Well, I appreciate you, Mark. And again, thank you for having me on your show. This is quite an honor to sit here with you in this space. So one day I'll come out to Puget Sound and see you out that way too.
Oh, I hope so. We'll do some art with luminous bacteria. It's just fabulous stuff.
All right, great. Definitely.
And I want to wish you and your family and your new laboratory group the very, very best.
Thank you very much.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with wonderful links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Nkrumah Grant is in the microbiology division at the University of Illinois at Urbana-Champaign. Fabulous place, by the way. Many thanks to David Renata for superb editing as always, and Reber Clark for the wonderfully quirky music that brings a smile to my face. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.